Hello, everybody. Thank you for the very nice introduction. I'm very happy that we can all see each other, if only in this virtual setting here. And let me start by sharing my screen with you. Here we go. Of course, it would have been much nicer to all get together and to have this lovely meeting in, looking for my pointer here, there we go, have this lovely meeting where we originally wanted to have it, and that is in Antwerp. And at least I was curious enough to see what Antwerp looks like and what Antwerp looked like a long time ago. And I found this lovely medieval map of the city, surrounded by its city wall with all the different gates and close to this river here where there's probably a big harbor. And I thought of this as, you know, as many medieval cities were, a combination of both protection of what's within the city walls, but then also, of course, intense selective communication with whatever is on the outside. And when I looked at this map a little longer, I realized that it really, really reminded me of my favorite organelle in the cell, which is, of course, the nucleus. And if we think about it that way, then the nuclear envelope, indicated here in this slightly liberal artistic way of my undergraduate student, also performs both of these functions, right? It protects the nuclear interior with the chromatin and the nuclear proteins against activities in the cytoplasm. And then it also has gates and openings here indicated by the nuclear pores, which allow for trafficking and for information exchange between the nucleus and the exterior. And I think this is probably the largest endoplasmic reticulum lumen that I've ever seen in any drawing. And I think it's, again, because Lily Thompson, my undergraduate student, who is an art and molecular genetics major, took some artistic liberties and made it quite similar to the river in the slide we just saw before. Now, this gets me all into the actual topic of my talk today, which is the function of the nuclear envelope in a eukaryotic cell, and specifically the function of the nuclear envelope and nuclear envelope associated proteins in plant cells. So let me start by taking you through a few slides that relate to this function. And the first one we all have in mind is, of course, what the nuclear envelope does is it contains chromatin. But it doesn't only contain chromatin, it also already organizes chromatin. And in this slide from a mammalian, from a mammalian nuclear envelope, this is indicated by showing a variety of different functional proteins which are embedded in the inner nuclear membrane and which have domains which reach out into the nucleoplasm. And here, for example, LBR, lamin B receptor, has a binding site for HP1, which is heterochromatin protein 1, and through this interaction can directly bind to silenced heterochromatic regions of chromatin. You also see indicated here the nuclear lamina, a very important structure in metazoan nuclei. And this is a proteinaceous meshwork which underlies the nuclear envelope and which again is in contact with both the nuclear envelope embedded proteins and also the chromatin associated proteins. So the nuclear envelope contains and organizes chromatin. The nuclear envelope is important for the regulation of nuclear size and nuclear shape. So only rarely, like in this C. elegans worm over here, do we have nuclei which are perfectly spherical, like the textbooks always imply. In most cases, this is not true. For example, in this human neutrophil, here you can already see this is a cell that crawls through dense tissue, and so just like the cell, these nuclei are highly pliable, and you can see all this reorganization of the nuclear envelope. And a similar almost disorganization you also find in fibroblasts of old people. So this is a skin cells of a 96-year-old, and you can see how they contrast to the skin cells of a 9-year-old. Quite a bit of aging process has occurred here, one might imagine. And up here, this nucleus, which you can compare to this one down here, is that of an approximately 9-year-old patient of progeria syndrome, which is this rapid onset early human aging disease. And you can see that the sort of disintegration of the nuclear envelope here has a lot in common with this biologically old person's nuclei. So the nuclear envelope affects nuclear size, nuclear shape. The nuclear envelope is also involved in intracellular nuclear positioning. I picked one example here from the mouse skeletal muscle. These are syncytia, right? So these are long stretched out cells with with a number of nuclei. And there is a subset of nuclei which are called the synaptic nuclei, which are sitting right underneath the synapses. They're held there by protein-protein interactions. You can see in this mutant that this protein-protein interaction is now disrupted. These nuclei become dispersed, and these kind of individuals, as in case of a mouse here, have problems with the branching of the phrenic nerve. So the position of this nucleus here is important, although the precise molecular mechanism is not understood. The nuclear envelope affects nuclear movement. This is shown here in an example of the Drosophila eye disc development. There is a stage where this nucleus has to migrate first to the basal end of the cell, then back to the apical end of the cell, and that triggers differentiation in the next stage. And when that is disrupted, again, in mutants over here, then the next differentiation stage can't occur, and that interrupts eye development in the fly. So a number of these phenomena, a number of these processes I just introduced you to are actually orchestrated by a specific protein complex, which is called the LINC complex, for linker of the nucleoskeleton and cytoskeleton. The LINC complex is a protein complex which is embedded into the nuclear envelope, shown here by the outer nuclear membrane and the inner nuclear membrane and the perinuclear space, basically the lumen between the two membranes. Now this consists of two types of proteins. There are the inner nuclear membrane SUN proteins or SUN domain proteins, and these are embedded with a single transmembrane domain into the inner nuclear membrane. They form trimers for all we know. They have what's called the SUN domain close to the outer nuclear membrane, and then this trimer of SUN domains interacts with the C-terminal tail of the second type of protein, the outer nuclear membrane KASH proteins. These are tail-anchored proteins. It's a very short tail. This is the interaction surface with the SUN domain proteins. And then they have a variety of different cytoplasmic domains which interact, for example, directly with cytoskeleton or with cytoskeletal motors. And you can imagine how that contributes to the function, for example, of nuclear positioning and nuclear movement. The SUN proteins also reach into the nucleoplasm, and there are interactions then with, like I just mentioned, the nuclear lamina and probably also chromatin-associated proteins. So my lab is a plant lab, and so we are interested in two different questions. One is, are there also biologically relevant nuclear movements and nuclear positioning events in plants? And if so, are there link complexes in plants? And if so, are these link complexes involved in these nuclear movement and positioning events? And eventually, of course, by what molecular mechanism? So the answer to the first question is already obvious from the literature. Yes, there is a number of these events. So also nuclear shape changes, which I don't even indicate in this slide. So nuclear movement, one of the most fascinating nuclear movements probably of any organism, or at least the most long distance, is the movement of the vegetative nucleus of the pollen tube through the entire elongating pollen tube, remaining close to the tip to the site of fertilization. In trichome development, the nucleus reaches a specific position here at that trichome branching, and if that is interrupted, for example, by actin depolymerizing drugs, then trichome branching is inhibited. Nuclei move to and away from fungal attack sites, and there's a difference in their movement, whether it's a resistant or a susceptible interaction. Nuclei can move, triggered by blue light in pavement cells, just like chloroplasts do. Nuclei can move and be positioned in root hairs, and nuclei can also react to a mechanical stimulus indicated here by this pipette tip on this root hair nucleus. So a number of interesting phenomena. Are there link complexes in plants? So to get you to this question, I have to take you a little bit back now into the history of the Meier lab, and I'll take you back pretty much exactly 20 years ago, when my lab was studying the RAN cycle, which is part of the regulation of nuclear import and export. And we were studying a specific aspect of that, and that is how this accessory protein here called RAN-GAP, RAN GTPase activating protein 1, is sequestered and anchored at the cytoplasmic surface of the nuclear pore, and specifically how is that done in plants. And the sequestering of this kind of protein in the cytoplasm and outside of the nucleus is important for the RAN cycle to drive nuclear import and export in all eukaryotic organisms. So at that time, it was known that in yeast, RAN-GAP is simply excluded from the nucleus, while mammalian cells had evolved a pretty specific mechanism. There is a unique C-terminal domain, which then gets stimulated. And that stimulated region attaches to a specific nuclear porin, which is on the outside of the nuclear pore at the cytoplasmic surface, and that then sequesters RAN-GAP there. So my postdoc at the time, Anne-Catherine Rose, was studying this project, quickly found out that plant RAN-GAP looked neither like yeast nor like mammalian RAN-GAP. It didn't have that stimulated C-terminus. Instead, it had a plant-unique domain at the N-terminus, and we named that the WPP domain after a conserved tryptophan proline motif in it. So Anne-Catherine showed that that WPP domain was both sufficient and necessary to anchor plant RAN-GAP to the nuclear envelope. So let me get back here. So that triggered my curiosity at the time, because I thought if plants have evolved a different functionality here for this very conserved, very basic plant cell biology, general eukaryotic cell biology question, which is how do I keep the molecule RAN-GAP in the place where it ought to be in the cell? If plants have evolved a whole different mechanism for doing this, then that must mean something, right? And most likely it means that the docking station for these molecules must be different in plants. So whatever this is not available. The way this works might not work in plants. Something else has evolved here that provides for the same mechanism. What is that something? So we went about to try to find what the interactors of the WPP domain is. My graduate student at the time, Zhanfeng Zhu, did the yeast to hybrid based approach, and we identified this family of 3 proteins, which we called WIP 1, 2, and 3. There's 2 splicing variants of WIP 2 here. And he showed that WIP1 bound RAN-GAP, all 3 WIPs bind RAN-GAP. This interaction is through the WPP domain on RAN-GAP and is through this coiled coil domain of WIP1, 2, and 3. And other than the coiled coil domain, these proteins have a potential NLS, and they have a transmembrane domain right at the C-terminus, so they are tail-anchored proteins. So we also showed that in the mutant, in the absence of WIP1, 2, 3, we lost specific nuclear envelope retention of RAN-GAP. Now, WIP1 is located at the nuclear envelope. We showed that it's on the outer nuclear envelope by co-localizing it with a protein that is sitting in the nuclear basket of the nuclear pore, so a little bit off the inner nuclear membrane. We also confirmed this by immunolocalization. At the same time, 2 other people in my lab had taken immunoprecipitation mass spec approach, my postdoc Jelena Burkochacic at the time and graduate student Chao Zhao. And so they went about just with the WPP domain, and they fished out what that bound to in a plant extract. They found a second gene family of 2 members we called them WIT1 and WIT2. They were similar to WIP1, 2, 3, larger. They also had a coiled coil domain, and they also had a tail anchor. And so now with these 2 stories together, we had plenty of potential RAN-GAP anchors for the nuclear envelope. So now I'm going to cut a long story short, always sad for those who have done all the work, and just show you the end result of what these 2 groups of people put together. And that was, there is actually a heterocomplex of likely 1 member of the WIP family, 1 member of the WIT family. We don't really know exactly the stoichiometry of this complex. WIP 1, 2, 3 are within this family redundant. WIT 1, 2 are redundant. complex is held together by interactions through these two coiled coil domains. WIP has a tail that enters the nuclear envelope lumen, WIT does not. The RAN gap interaction onto this complex is through the coiled coil domains, through the WPP domain of RAN gap, and either in the absence of WIP 1, 2, 3 or WIT 1, 2, this falls apart and RAN gap becomes delocalized from the nuclear pore. So we were quite happy with that part. We still didn't really know why this is so unique in plants. Now, around the same time, and I think it was actually in SEB conference 2, where we first started to talk, these two wonderful individuals, David Evans and Katja Graumann from Oxford Brookes, had also started to get interested in the plant nuclear envelope. They had done a couple of different projects, but among them was that they got interested into the LINC complex, and they found by sequence similarity plant SINE1 and SINE2 in Arabidopsis, so the inner component of the LINC complex. But they and others could not identify any KASH proteins because nothing in the plant genomes looked like the animal KASH proteins. So we got together, and at that meeting we thought, well, I don't know, this is actually exactly in the right place and has the right conformation to act as a plant KASH protein. So I was fortunate to have an extremely courageous graduate student in the lab at that time, Zhao Zhu, and he took this project on. And again, I'll just click and show a lot of work here, but he showed that indeed WIP and SUNS interact. The interaction goes through the tail domain of WIPs. WIT does not interact with SUN, but interacts with WIP, since it's therefore in the complex. And in the absence of SINE1 and SINE2, WIP is no, and by extension, WIT are no longer associated with the nuclear envelope, and then neither is RAN gap. So we had, by all means, we published this in this paper indicated in red down here, we had by all means identified what we thought of as the first KASH complex known in plants. And we were fascinated by the idea that the inner nuclear membrane protein is quite conserved to that in animals. There's also some fungal ones now. And the outer nuclear membrane components are quite different. And so again, something is still happening here with plants having recruited through evolution different types of proteins into this functionality. All right, so here this just shows that the C-terminal tail of WIP1 is unlike that of the canonical KASH proteins from animals. It has this is the transmembrane domain, has a very short tail domain, and then not much sequence similarity to what was known. So the canonical tail here was really PPPT, and the plant protein doesn't quite have that. So Chow then thought, well, maybe plants among themselves have a different consensus. And so he aligned WIP-like proteins from a variety of different plant species, whatever we had at the time. And he found that that was indeed the case. So there's this short tail, which is conserved. Transmembrane domain, of course, is conserved. And then the plant proteins all had this valine, proline, threonine at the very C-terminus. So now we had a piece of information to use, and what he did was he wrote a brief algorithm which was looking now through plant databases with exactly this feature. Give me a transmembrane domain, give me a tail of unknown sequence of a certain length, and then give me a VPT. And so by doing that, he found in Arabidopsis 4 additional candidate proteins for plant KASH proteins. We found a 5th one in Medicago, and then again, it's a cartoon. He went through all the work to show that they all are on the nuclear envelope. They all are on the nuclear envelope via their tail, especially the VPT and especially the PT are important for their localization here. None of them is at the nuclear envelope in the absence of the SUN protein. And so by all means, they are, you know, again, by function, canonical KASH proteins. So now we had populated the nuclear envelope with these proteins. And at that point, my lab decided that what was really important was to figure out what their role, what their function really is in plants. And so I'm going to show you two stories today. The first one will be to go back to our original protein complex, but now forget about the role of RAN gap anchoring for a moment. And we found a phenotype of these proteins in male fertility. I'll tell you that story. And then we're going to focus on these two new proteins over here, SINE1 and SINE2. And these are actually related, 80% sequence similarity. SINE3 and SINE4 are unrelated proteins. I won't tell you anything about them today. And we discovered that these two proteins have a role in stomatal dynamics. And for all of those in the audience who are not in the plant field and for whom this is not like immediately obvious, male fertility, of course, translates into seed set, and seed set, as agriculturalists like to call it, translates immediately into grain yield. And stomatal dynamics is the way of plants to cope with specifically abiotic stresses. So here we're directly talking about drought and heat tolerance. And I think these are two topics of increasing importance in our days. Okay, so male fertility first. So I have to get you into pollen development in plants. So we go through meiosis. A male myocyte goes into meiosis, and out come 4 microspores. Now each microspore becomes a unicellular pollen grain that goes through a first round of mitosis, which creates the original germ cell, which is engulfed into the vegetative cell. So this nucleus is now also called the VN, the vegetative nucleus. That first germ cell develops a cytoplasmic extension that wraps around the nucleus, goes through a second round of mitosis, and then we have the 2 sperm cells attached to the vegetative nucleus. When the pollen grain germinates, this becomes a unit to travel. So this is also called the male germ unit, and I might sometimes say MGU, male germ unit. And this unit now travels through the elongating pollen tube to eventually reach the female gametophyte, where the fertilization of the egg cell in the central cell will occur. I'll show you that in a second in a different cartoon. The 2 sperm cells remain attached to the vegetative nucleus, and from the studies that were published before we started this work, it appeared that the vegetative nucleus is the driving force of this whole unit. So we had found in these mutants, both WIP and WIT, that they have a seed set defect. So they have about 50% reduction in seed set, and it comes from the male parent. So we wanted to know what their localization in the pollen grain is, and we found that both of these types of proteins, I'm showing you one example each here, are predominantly localized at the nuclear envelope of the vegetative nucleus. There is very little of any staining of the germ cells here. So what's going on in the mutant then? So here we have now labeled the 3 nuclei, the vegetative nucleus in red and the 2 germ cell nuclei in green. And so this is the picture you get early after pollen tube germination in wild type. You see all these packages of the 3 nuclei with the vegetative nucleus ahead at a certain distance from the tip. And if this was a movie, you would see it slowly crawl towards that elongating tip. In the mutants, both WIP and WIT, I'm showing one example here, we see a different picture. We see, first of all, that the order is changed already early on. So the germ cells are transported first, the vegetative nucleus trails behind, but then if you follow a couple of them, you can see that they also get separated. So here's a number of pollen tubes which apparently have left the vegetative nuclei already far behind. And then there's a number of pollen tubes out here where you can see only the germ cells are migrating. And this is comparing this now in a short movie, so you can see the normal trafficking pattern here in wild type. On the left, the unit is crawling along, and then if you pay attention here on the right, you'll see how the distance between the germ cells and the vegetative nucleus is already stretching out in this brief movie here. So when we looked at later stages, now we're looking at 16 hours of semi-in vivo germination. We have labeled everything in green now. The reason for that is we also want to see the cytoplasmic extension. And so you can see in the mutant, we find cases where the vegetative nucleus is trailing behind. This extension is becoming very stretched out. And we see cases where they have parted quite at a distance. The extension has ripped probably at this point. And then we see other cases where a callus plug has been formed. So this vegetative nucleus is now completely blocked from further continuing the tip growth of the cell. And we quantified this. About 50% of the WIT pollen tubes had these defective vegetative nuclear migration events. So what happens at the other end? So what happens during the fertilization process? Normally, the pollen tube reaches the female female reproductive structure. And then the final stage of a complex multi-stage process of how it gets there is actually called a pollen tube termination. And it's simply a burst of the pollen tube, and that releases the two sperm cells. Not really clear what the vegetative nucleus does here at this point. The two sperm cells will then fuse with one with the egg cell that will produce the embryo, and the second one with the central cell, and that will produce the nourishing endosperm. Now, if that process is disrupted, specifically that pollen tube termination process, so the burst, you can monitor that by watching out for two phenomena. One is pollen tube overgrowth, and that simply means that that non-bursting pollen tube keeps growing inside the female apparatus. And the second one is polytubie, because pollen tube burst sends an inhibitory signal for a second pollen tube to arrive. And when that inhibitory signal is missing, then you can sometimes see two pollen tubes arriving in the same place. So we watched out for this in our mutant, and we did this by labeling now the cytoplasm of the pollen tube in green, and then you can see the egg cell nucleus here in red. And so in wild type, what you see is a brief burst and a diffuse GFP signal when that burst has occurred. When it doesn't burst, you see these stalled pollen tubes, like in this slide here. Overgrowth is quite obvious because the green fluorescence signal is then migrating further inside than it normally would. And in some cases, we could also trace down polytubie, so we can see two different pollen tubes here. And so the combination of stalled and overgrown pollen tubes, in contrast to the burst pollen tubes, was roughly 50% in this experiment, and that matched very nicely both the seed set defect, which was roughly 50%, and then maybe I haven't mentioned this earlier, in the pollen tube termination and growth experiments, we also had about 50% mutant pollen tubes, which had the vegetative nucleus left behind and not migrating with the germ cell. So all these things happen in half of our mutants, which is simply what we at least think of this simply as the penetrance of the mutant phenotype. Okay, so we know at this point that these KASH proteins have this effect on a pollen tube termination. Our hypothesis is that the reason for this is that there is an important function of the vegetative nucleus at the very tip of the pollen tube at the time of termination. So what is that possible role? So we have a hypothesis for this, and I'm only going to give you a brief overview of the hypothesis. Most of the regulatory factors for pollen tube termination that are known in the literature are about the female, and little is known about the pollen. So what is known is that at the time of pollen tube arrival, I really thought I turned everything off here. At the time of pollen tube arrival, the synergid cells react with a burst of reactive oxygen species. That leads to an increase of tip calcium in the pollen tube, and that calcium burst migrates into the flank of the pollen tube, and together these phenomena are involved in burst of the pollen tube. So what we hypothesize in our mutant now is, what we know is that there is this reversed order, and importantly, I think, is there's this increased distance between the vegetative nucleus and the pollen tube tip. So what we think is that the Ross burst still occurs, that somehow this nucleus at a distance is now no longer able to pick up a signal at the pollen tube tip, and this signal is most likely the calcium influx, and that therefore downstream of this event, the burst can no longer occur. So we're focusing in on reactive oxygen species and calcium here with our hypothesis, and that's all I'm going to tell you right now. What I do want to tell you is that you should also attend session C2 on July 6th at 2 p.m. to 2:30 p.m. British summer daylight saving time, I believe. And that I will tell you how these two graduate students in my lab, Andrew Kirkpatrick and Morgan Moser, have actually picked up this hypothesis and what their findings were. All right, with this, I would like to switch to the stomata part of the story. And here we're switching also to different KASH proteins now. We're going to talk about SINE1 and SINE2. So what do we know about these? By the way, we call these sun domain interacting nuclear envelope proteins. So just like WIP, they are plant-specific KASH proteins. They're both tail-anchored. These don't have a coiled coil domain. They have N-terminal armadillo repeat-like domains. We have shown that SINE1 co-localizes with F-actin, specifically the N-terminal arm domain co-localizes with F-actin. That's what's indicated in this cartoon up here. SINE2 does not. In terms of expression, this is how we got into GARD cells in the first place. SINE1 in leaves is predominantly expressed in GARD cells. Both proteins also have a root expression pattern. SINE2 is expressed in mesophyll and pavement cells and only weakly in GARD cells. And I think I mentioned this before. This is a gene family of 2 members, 80% amino acid similarities. So again, what are GARD cells, right? GARD cells are the 2 cells that form the stomata in plant leaf epidermis and other epidermal tissues of the plant. And these are the microscopic pores, which are important for gas exchange, CO2 against oxygen, and also for the release of water vapor. And they are tightly regulated in response to a number of environmental and internal factors. And one of the most well-studied regulators is abscisic acid, one of the plant hormones. And this is the response to drought stress. So in response to drought, abscisic acid increases in the plant lead from this open configuration of these 2 cells. This is all regulated by a vacuole turgor, right, and ultimately by ion channels in the plasma membrane, and leads to this closed conformation, and that is simply protecting the plant from excessive release of water vapor, but at the same time is also slowing down then photosynthesis. And it's a whole world of plant physiology, which we can't talk about today. So, okay, back here. So Alicia Beer, my graduate student, took on this project and decided she really wants to know whether there is a role for these proteins in this process, and did her tests. And yes, indeed. So we see that if we stimulate these epidermal peels with ABA, in wild type, the GARD cells are closing after 3 hours in this assay, while the mutants are remaining partially or mostly open. And we can complement this by putting mini genes of either SINE1 or SINE2 back into the plants, and then the mutant defect is affected. So both proteins, the one that's highly expressed in GARD cells and the one that's weakly expressed in GARD cells, have a defect in stomatal closure. So a number of molecules are known that act downstream of ABA in this process. I'm going to focus on 2 here today: cytoplasmic calcium and the actin cytoskeleton. So it's known that release of cytoplasmic calcium, which is oscillatory, is involved in ABA-based stomatal closure. And it's also known that these oscillatory calcium spikes are then decoded by downstream molecules, which eventually lead to the activation of the mentioned plasma membrane inserted ion channels. And on the other hand, it's known that the actin cytoskeleton is reorganized in a spatiotemporal manner during closure. And it's also known, some indirect evidence, that the organization of the actin cytoskeleton feeds back on the activity of these ion channels. It also feeds back on the vacuolar morphology changes which accompany GARD cell closure. So all of this has been known from independent papers by a number of different groups, some of them mentioned down there. Okay, so we wanted to know how our mutants relate to all of this. Our first experiment was to test the mutant phenotypes if we stimulate GARD cell closure. If we stimulate our GARD cell closure with calcium instead of ABA. And what we see here is that calcium, at least partially, but quite significantly rescues the mutant phenotypes, both for SINE1 and for SINE2. So calcium is known to be downstream of ABA. This places in our mind our mutants, our proteins between ABA and calcium in this pathway. So the other factors, a little bit more complicated to explain, is actin reorganization. So this is what's actually happening. What I mean with that, the wide open stomata have their cortical actin organized in this what's called a radial affecting array. So you can imagine, you know, how they crisscross the guard cells in this pattern shown here. When they close, they go to this transition stage where apparently most of the F-actin or an amount of the F-actin depolymerizes, and you either see a diffuse signal, which is whatever fluorescent marker you use, or this reduced mixed pattern of shortened F-actin. And then in the fully closed guard cells, they are re-polymerized into these longitudinal actin cables. So this is what's undergoing in wild type. So first, we were interested in seeing whether our mutants and actin depolymerizing and actin stabilizing drugs would interfere. And it was known in wild type that there is some activity of these drugs, and specifically of an actin stabilizing drug. I'll tell you that in a second. So first we used the actin depolymerizing drug, the tranquilin B. That has little effect on wild type. Possibly it enhances closure a little bit. That's those two lines to compare. Now what we know is that the SINE1 mutant with ABA, that's our green dotted line here, has problems closing, but then adding the tranquilin B significantly improves this closure. So just like calcium can rescue this mutant phenotype, the tranquilin B can also at least partially rescue the mutant phenotype, indicating that our mutants are somewhere in the actin pathway. And the same is also true for SINE2. Now with the actin stabilizing drug, JK, there's a different effect. So first of all, there is an effect on wild type here. So wild type closes in the presence of ABA. Wild type is inhibited from closing in the presence of ABA and JK. The SINE1 mutant cannot close in the presence of ABA, the green dotted line. However, in the presence of ABA and JK, closing is induced, but interestingly, in the presence of JK alone, this mutant is now also closing. So actin stabilizing drugs in combination specifically with the SINE1-1 mutant leads to closure in the absence of ABA. This effect is not true for SINE2. This is the first time we're seeing a clear difference here in the role of these two proteins. So again, wild type is inhibited in closure. SINE2 never closes under any of these treatment conditions. Okay, so then we wanted to know what is actually going on with the actin cytoskeleton now under these conditions. So we introduced GFP Live Act as our green marker here. And the top panel shows you what I've already told you. So we go from this radial organization to a largely diffuse and mixed organization to these longitudinal panels. And we quantified this over here. So we call radial type 1, it's the green bars. The diffuse actin is the light blue. The mixed actin is the dark blue, and then the longitudinal bundle is the purple. So you see that wild type goes from mostly radial to mostly diffuse mixed to mostly longitudinal. The SINE1 mutant, and I think you can also clearly see it in the example panel over here, never has a proper radial organization in the open guard cells, then reaches this mostly diffuse depolymerized stage, but then stays there, never is able to repolymerize actin at least at the end of this assay. And the SINE2 mutant does something altogether different. SINE2 mutants reach this disorganized state. So you can clearly see in the panel here that this never becomes diffuse and actin never properly depolymerizes. So reaches a disorganized state, already starts less radial than wild type, and then remains in this disorganized state. So SINE1 has a problem with repolymerizing its filaments, and SINE2 has a problem with originally depolymerizing its filaments. That's how we interpret these data. And we did a number of quantification assays measuring all the angles and so on, which I don't have time to show you today. So we knew about the calcium effect. We want to know, can calcium rescue now this effect? And as always, our calcium rescue remains partial, but we think we can, just like the closure rescue was partial, we think we can interpret these data. So in wild type, calcium leads to some of the expected reorganization. So it goes from type 1 to type 3. It's less less complete than in wild type, just like closure was less complete. And then in the mutants, we also get a partial rescue. So remember, SINE1 was mostly depolymerized, light blue, and now SINE1 has all these mixed states, but importantly, it reaches to a certain percentage the type 3 state. And SINE2 was mostly dark blue in the ABA assay, and it also goes through less dark blue here, and it reaches the purple a longitudinal state. So we interpret this, and again, we quantified it as a partial rescue of the actin reorganization by calcium, which again places our SINE proteins, and I should have said this earlier, it places our SINE proteins upstream of the actin reorganization, but it places calcium between the SINE proteins and the actin reorganization from these data. So this is a summary. This is what we think happens, right? So wild type depolymerizes and repolymerizes F actin. SINE1 depolymerizes but can't repolymerize, and SINE2 can never properly depolymerize in the first place, and calcium partially rescues both defects. So now with that, we know that the SINEs are upstream of calcium and of actin. Calcium is upstream of actin. So now we really wanted to know what the effect of our SINE mutants would be on the cytoplasmic calcium fluctuations. So this is normally what happens in wild type after you trigger these guard cells with ABA. You see this spiking over a period of 5 to 15 minutes. And these oscillations is then, like I mentioned before, they're decoded, and they eventually are required for the activation of the ion channels. Okay, so we try to recapitulate this type of experiment and then compare wild type and mutants. This is the work of my current graduate student, Morgan Moser. She decided to use the ratiometric calcium sensor YC3.6 for this experiment, and we've used a nuclear export signal to it because we want to really only look at cytoplasmic calcium in this case. This is just a false colored example here of what this first peak and this trough looks like in this experiment here. So she can recapitulate the typical calcium spikes in the cytoplasm in wild type. She also sees them in the SINE1 mutant, but they are mostly absent in the SINE2 mutant. And this is all quantified here over on the right. So again, like before in the JK experiment, we see a clear, and in the actin organization experiments now, we see a clear distinction between the mutant phenotype of SINE1 and the mutant phenotype of SINE2. So our next question was, of course, what rescues this? And so we can rescue this with external calcium. So she adds external calcium instead of ABA. SINE2 starts to fluctuate again. So these are no longer statistically different from each other. And when she adds LAT-B, interestingly, so now she depolymerizes the actin cytoskeleton, she can also rescue the SINE2 phenotype. So allowing for the depolymerization of actin by the drug, which in the SINE2 mutant is inhibited, rescues the calcium spikes in the cytoplasm. So again, in this case, placing the actin depolymerization step, which is inhibited in SINE2 upstream of the calcium fluctuations, thereby providing a possible explanation for why we see the calcium fluctuation phenotype only in the SINE2 mutant and not in the SINE1 mutant. So my lab is in general also interested in nuclear calcium. You'll see more about that also in the short pollen tube talk. And nuclear calcium fluctuations have been shown to be important in a variety of other biological phenomena, biotic and abiotic stress, root development. There are more and more examples coming. It hasn't been investigated for stomata. So we were just interested, given that we are working on nuclear envelope associated proteins, we thought it was a short step to also ask whether anything in this particular biology is going on with nuclear calcium fluctuations. So Morgan went and took a different sensor. Here we made use of an NLS fused R-gecko. So this is not a ratiometric, but a colorimetric calcium sensor. So you see either brighter or dimmer red fluorescence in this experiment over here. The other one was a FRET-based assay. I might have forgotten to mention this. And so first of all, yes, ABA can induce nuclear calcium fluctuations. They are less regular than the cytoplasmic ones. If you compare them on a time scale, they're also not synchronous with each other. They're also fewer, but we quantified all of this nevertheless, and she found the same phenomenon she had found for the cytoplasmic calcium fluctuations. There is no distinguishable defect in SINE1-1, but there is a clear defect in SINE2-1. So at this point, we know nothing about the biology, the potential regulatory features of these nuclear calcium fluctuations, but because they hadn't been known in the guard cell biology story before, we're quite interested now in also following up on this to a certain degree. Okay, this leads me to my current model of this story. And for me personally, the most important part of what came out of all these data is that there is a clearly distinguishable role between SINE1 and SINE2. They're acting in the same pathway. They're both required for stomatal closure, but they're required for stomatal closure in quite distinct ways. So we think that SINE1's main role is in some way to assist in the actin repolymerization stage of the actin reorganization. This can be rescued by external calcium. So calcium is certainly involved in a certain degree, and this is required for stomatal closure. Now, in contrast, SINE2, as we saw, was more involved in the earlier actin depolymerization stage. And we have clearly seen through the LAT-B experiment that actin depolymerization is required for the internal calcium fluctuations, and SINE2 in turn is required for the internal calcium fluctuations, but SINE1 is not. Both the actin depolymerization and the internal calcium fluctuations can be triggered when we add external calcium. And SINE2 is also required in triggering internal nuclear calcium fluctuations, which can also be rescued in the mutant by external calcium, and how that part of the pathway fits in with anything else. And we don't know, and specifically you will notice I have not drawn an arrow to stomatal closure because we have no idea if this is involved in stomatal closure at all. So, but this is pretty much where we are at the stage. This is a paper currently under review. And of course, our next stages will be up here. We really want to know in what molecular way are these proteins involved in triggering all these events. That gets me back to my very original slide of this talk. And that is, I think I've tried to give you an overview of the role of the nuclear envelope in general in eukaryotic organisms. It's more than a bag that holds your chromatin in a tight place. And specifically, I've tried to give you an overview of A, how we got into this story, and B, how we are interested in dissecting specifically what's going on at the plant nuclear envelope and what protein functionalities are involved in this second role, which might be the more important role of the nuclear envelope, and that is its ability to help communication between the nucleus and its cellular environment. So just as a little reality check, all I've given you is 3 more proteins, or 4, which are anchored into the plant nuclear envelope. And I told you stories about their role in male fertilities and stories about their role in guard cell regulation, 2 very important pieces of plant biology and plant physiology. But as you can imagine, there are multiple question marks for us to address now. And for me, most important is we need to understand by what precise molecular mechanism are these molecules performing these important roles. And that, of course, gets us into understanding what are their protein interaction partners, what is their molecular role, what is the molecular role of the protein interaction partners, how is this all connected. And for the SINE1 and SINE2 story, more than for the WIT story, you will have noticed that if you followed all along, is how is any of this I just told you connected to the role of the nucleus in guard cells. And I think that is the big question that will tie us back to the whole link complex and KASH protein story, which is how we got into all of this. Okay, with that, all that's left is thanking my lab. And I think I have mentioned everybody along the way who has been and is still involved in these projects. There are a couple of people in these pictures that you probably haven't seen. Anna Newman has done a beautiful paper a few years ago on the link complex in Medicago, because we also wanted to branch out into an important crop plant. Norman Groves is my lab manager, super postdoc, and involved in almost everything I have talked about. He also has a second talk, probably had a second talk already, on his interest in internuclear membrane proteins, and thank our lovely collaborators, David and Katja, and of course, as always, my main funding source, the NSF. So I'd be very happy to take questions.